we have Dr. Itamar Kahn. Itamar did his PhD already in MIT, Boston, where he studied cortical contribution to episodic retrieval. Did it in Anthony Wagner's lab. So this postdoc, he moved not so far away to Harvard University, actually to the hospital, so it's farther. Did their functional connectivity in monkeys, and he later developed the opto-imaging technique, or opto-fMRI technique, to manipulate neurons and then follow their change in activity and how it affects behavior and neurodegenerative diseases. And then he opened the lab in Technion. Today he's going to talk about corticostriatal disruption in NF1 revealed by functional connectivity MRI in awake mind.
Thank you, Amit, for the kind invitation and introduction. So, I contributed to optogenetic fMRI, but I refuse to call it a novel method. Taking two things that work and just doing them together is, I don't know, it's just doing them together. So, but we did optogenetic fMRI, and the goal, which is something that for me was a precursor for doing what we are doing in the lab, was really using fMRI as what I like to term a translational bridge. So being able to kind of compare between the species, between animals, mice in my case, and humans. I think it has relevance both for understanding disease. So as I'll show in the second half of the talk, animal models of disease, trying to understand and even try to identify potential target kind of mechanisms that are relevant for humans. And just by the mere fact that we use the same exact method to image the brain, you can compare across the species. And actually what I've done during my postdoc was to demonstrate that the hemodynamic response that we see in humans, which we evoked it with optogenetics and with whisker deflection, so sensory stimulation, is equivalent. So the parameters of the hemodynamic response function are equivalent, and it can be used in a meaningful way. So this is kind of the logic behind that. There's one additional aspect which is worthwhile when you think about fMRI in small animals, is the fact that you can image the entire brain at the same time. So this is something that it gives you relatively for free. So that's along with the other limitations of fMRI. Okay, so the lab is interested in understanding the basic organizing principles of functional networks in the brain. This is showing the phylogenetic tree, and really we are looking at the interaction between networks that underwent, or systems that underwent significant expansion and elaboration, like the neocortex, and other subsystems that were relatively conserved across evolution. So cerebral cortex relative to the hippocampal memory system and the basal ganglia. Two systems that we are interested in, and we're interested in the interactions between these systems. So the first half of the talk, I'll describe our work on the hippocampal memory system, and in the second one, on the basal ganglia. We are imaging in our facility mice. In collaborations, we are imaging humans and monkeys. So the monkey work is with Wim van Duffel over at Leuven. Okay. So these are the two parts of the talk. The first part, I'll talk about the method in general that we use to map these brain systems, and I'll describe the cortico-hippocampal connectivity. And in the second part, I'll describe an animal model, neurofibromatosis type 1. It has many effects. I'm focusing on its effects on the nervous system, the central nervous system, and I'll talk more when we'll get to that. Okay. So cortico-hippocampal connectivity, and then abnormal corticostriatal connectivity, and also at the end I'll kind of link it a little bit to current ideas about autism, which has high incidence in neurofibromatosis. Okay, so from the method's perspective, I'll just really show you one slide of an awake mouse imaging apparatus. Then I'll spend most of the talk describing results using intrinsic spontaneous activity, and I'll define exactly what is that. And at the end, if time permits, I'll show you our awake behaving fMRI for mice with virtual reality and animals running on a treadmill and all the different little neat things, and olfactory stimulation and that we got. Okay, so awake mouse imaging. Again, here we didn't invent anything. We took everything that was proposed with two-photon imaging and just applied it to a MR-compatible setup. So here what you're seeing is actually a mouse that is already being trained to take water from a little water tube. We actually pulled it a little bit backwards, so you will use its paw to reach and collect the water. You can put it closer, and then it will collect it with its tongue. What's important here is that we acclimate the animals to be in a head-posted position by simply attaching a little plastic rod to their skull. It's a standard procedure for acclimation, between 3 and 5 days of increasing amounts of time. The animals learn how to do that. They learn that the whole situation is, mice are stressogenic. They become stressed by almost anything. They are stressed, but we see that their stress levels go down. They don't go to baseline levels, but they are relaxed enough, so they'll be about 30 to 40 minutes in this head-posted position. It's important to note that we are not holding their body. So their body, they can actually move it if they want. They don't have enough force to actually break from the head post, but they can move their body, and when they become restless, we see that about 30 minutes, they will become restless. So a little bit like humans, 30 minutes in the scanner, they had enough, they'll move, and we are taking them out. So this is what we get with these animals. So in this case, if you want to teach them to do the essentially perception with water, we do water deprivation. We worked a little bit with soy milk, like others in the field are doing. It kind of works. They'll actually do without water deprivation for soy milk a little bit of work, but you cannot have a prolonged behavioral trials like you have with the water. So if you do water deprivation, it's much, much easier. For the experiments that I'm describing here, we didn't deprive them of water at all. The only thing that they do like quiet wakefulness. We hold them in a head-posted position, and they're not doing anything. So it's like the equivalent for human resting state experiment, where the person is lying in the scanner, staying still. Okay. So intrinsic spontaneous activity. I'll describe the phenomenon in humans, and then say a word about animals. So this is from Bharat Biswal, showing one of the first demonstrations of this phenomenon. Essentially, what Bharat Biswal noticed is that if you take a subject, it's a human brain, it's over anatomy, you put the regions where if they move their right hand in red or their left hand in blue, you get activation in the primary motor cortex. He noticed that if you actually look at the time course of the fMRI signal when the subject is not doing anything, you're going to see highly correlated fluctuations of the fMRI signal between the left and the right hemisphere. You can see from this that the half cycle of these fluctuations is about 15 seconds, so very slow fluctuations of the bold or blood oxygenation level dependent fMRI signal. And moreover than that, if you actually take one of these time courses and ask where else in the brain do we get these spontaneous fluctuations, you actually get the entire, or what seems to be the principal components of the motor network in the human. Motor, primary motor, SMA, supplementary motor area, S2, striatal regions, even cerebellar regions. And many, many studies have been done using this method, both on the motor system. These fluctuations going on in the entire brain. They go on in all of the networks, and essentially they allow you to identify functional networks in the brain. So just a summary of the results. There are literally hundreds of papers using that method in humans. There's also evidence in monkeys, rats, and mice that it exists, and I'll show you work in mice. You can see all of these different networks. It exists in the absence of explicit input or output, so the person can be not doing anything. If they actually do something, these fluctuations will not go away. They might be attenuated or even exemplified, increased, but they're not going away entirely. It does not disappear during task conditions. It does not disappear if the person is asleep or anesthetized. It's simply being attenuated. So it's there. It's similar brain networks across species. So we kind of would like to claim that it's actually a very effective way to compare across species, right? If we have the same phenomena in different species, if we actually don't engage, we don't need to use a task to compare between the species, it might be even better to look at networks where there isn't a very simple homologue task that you can give across the species. Okay, so this is a human, one hemisphere, cerebral cortex. This is just one scan where you simply plot these time courses for each point in the brain. You speed it up a little bit. Yellow, red means high, blue means low. If you look at, say, these frontal parietal regions, you can see that they kind of go up and down together. The visual cortices, if I would have shown you the other hemisphere, its visual cortices would go up and down together. And a mouse brain, so drawn to scale, and then if you do the exact same thing, you see the exact same phenomenon. I'll show you in a bunch of analysis that this is something that is highly replicable across animals. We've done a lot of work on that, and you can actually study the organization of the system using this method. Okay, so intrinsic signals can be used for functional mapping and cross-species comparison. Okay, so I'm going to highlight, so we are really looking at these different species, some work I've done before starting my own lab. Today I'll highlight 3 of the studies that we've done in the past few years. Okay, so the basic setup is an awake mouse. Now it's a top-down view, so this is the cradle. To make it MR compatible, we just use a 3D printer to print it out. If anyone is interested in a cradle like that, I can send them the files and they can print their own. It's actually very convenient to work with these printers, and you can adjust the settings. Here you can also see a single element, 20 millimeter receive-only loop coil that we use to image the entire brain. All of the functional data that I'll show you is 150 microns in-plane resolution and 450 microns slice thickness. So you can take these functional images. So the way we compute it is we take a region of interest and we compute all the other voxels in the brain that show correlated activity. We do some pre-processing that is conventional in the field. Afterwards, if someone is interested, I can tell you more about it. We actually align it to the Allen Brain Mouse Atlas and the Allen Brain Mouse Connectivity Atlas, and I'll show you some analysis that we do using this connectional anatomy data. So this is actually the EPI data, so it's high resolution enough so it's not distorted. We get an image of the entire brain, and then we can reconstruct it and put it on a surface and look at regions on the surface of the brain. So this is using FreeSurfer, the same tools that folks are using for humans. Here is just an example of signal to noise in a region that we have a lot of interest in, the post-rhinal, perirhinal, and entorhinal cortices. This dropout that you see in the signal is due to the ear canals. So same susceptibility artifacts that you get in humans affect equivalent regions. We can do more than that. So again, this is the EPI data. We can demarcate the subfields of the hippocampus, and we can actually reconstruct each one of them separately as a 3D structure, and then we can flatten it and project the data onto this flattened surface representation. And it's accurate enough such that if I show you a map and you see some foci of activity, you can take the bregma, essentially the Paxinos and Franklin Atlas coordinates, superior, inferior, anterior, posterior, and stick an electrode right in that region. And this is something that we are also doing. Okay, so one of the questions that we had and we asked was what would be the sensory representations in the mouse hippocampus. There is evidence from the literature that sensory responses are observed in the mouse hippocampus, and we were wondering whether we are going to see an entirely integrated responses of sensory regions in the hippocampus. And in order to ask that question, we planted seeds in the primary somatosensory barrel field area, primary visual cortex, and the primary auditory cortex, and we computed correlations in a group of animals. So the group of animals, these are 12 animals. Each one of them is scanned between 5 and 10 scanning sessions. A scanning session is 1 day where we scan the animal. Each scanning session is about 32 minutes. The total is 100 sessions, which is the equivalent of putting 300 human subjects in the scanner. So this is a very extended data set that we acquired. What you can see here is the three primary sensory regions for each one of the hippocampal subfields. And what you can see is that you get these statistical parametric maps of the group data for each one of the subfields. You can see that here in the dentate gyrus there seems to be a kind of spatial segregation between the different senses. If you go to CA3, there's slightly more overlapping, CA1 slightly more overlapping, and subiculum more overlapping. We quantified that those responses, and we've seen, so first in terms of the coverage in each one of these regions, we see that their coverage is relatively stable, but still we see that they become more and more overlapping. So we essentially computed unimodality, which means that the voxel is not overlapping with any of the other modalities, versus heteromodality. So there's overlap between the voxels. And what you can see here is that between the dentate gyrus to the subiculum, there's a decrement in the unimodality. And between the dentate gyrus and the subiculum, there's an increase in heteromodality. So essentially, gradually, more and more overlapping representations, suggesting that there's some integration going on in the hippocampus proper, and again, pointing out that unlike conventional wisdom, it seems that there are some biases within the dentate gyrus for a modality-specific representation. So again, this is something that it's a clue to now test with electrophysiology. It should be noted that people have observed sensory-specific responses in the hippocampus. So we are with electrophysiology, so we are not the first to note that effect, but this is the first time it has been shown with imaging. Okay, so this analysis is actually just showing you, for the CA3, sorry. Yes. So now we are working on that. That's, that's the next part is actually to use visual stimulation, somatosensory, and we have olfaction, which is, I'm not showing you the analysis here. But, this is, we want to do more than just show passively information. So we actually want to do like hippocampal dependent learning. So. But this image is meant to show is this is each one of these graphs is each one of the individual 12 animals and the average response, showing the foci of these responses along for the three different senses, showing that overall across the different animals we see relatively a stable response. So it's not just one or two animals showing this peak response in that region, but really it's something that is consistent across animals. Okay. And the last analysis that I'll show you from that work is actually comparing not just the primary sensory regions, but comparing representations across the entire cortex. So each one of these seeds we computed, it shows up here in color, the percentage of coverage in the hippocampus, in the parahippocampal region, in mice, and in humans. And what you can immediately see from this analysis is that there's essentially a flip between the representations in humans and in mice. So while in mice there's a very significant representation in the hippocampus for primary sensory regions and high order sensory regions and very little for association regions, in the humans the hippocampus is dominated by association regions and has virtually no representation of sensory regions, and the parahippocampal is in between. So how do we interpret that response, that flip? So this is from George Streeter, the representations of interaction between the neocortex and the entorhinal as the entry point for the hippocampal memory system. What we'd like to argue is that while in rodents there's strong projections, and we know that there are projections to the parahippocampal regions of sensory region, of primary sensory information as the entry point to the hippocampus. Association regions play a very, like a much more minor role. In humans, there's been a rerouting event occurred. Essentially, all of the information that goes to the hippocampus goes through association regions. So almost no input from these regions to the hippocampus, and everything going to the association cortices. The implication of that is essentially abstract memory representations are the type of information that enters the hippocampus, and very little primary sensory and even secondary sensory information enters the hippocampus. Okay. So I hope that this was kind of an example of how we can use this spontaneous activity to do mapping of these systems, even identify novel patterns. The comparison across species reveals conserved basic organization of cortical networks. I haven't shown you that, but we show also relative to projections, anatomical projections in the alien brain, that the functional connectivity mimics them in the mouse very, very well, but we see dramatic rerouting of information. And now we ask ourselves, can we use this type of approach to identify potential disease mechanisms? Okay, so neurofibromatosis type 1, it's actually a fairly common condition in Israel. It's a neurodevelopmental condition, one out of 1,000 in Israel, autosomal dominant. Neurofibromin is mostly in neuroglial tissue. The lack of neurofibromin results in RAS hyperactivation, increased cyclic AMP, PI3K, induced hyperactivation of mTOR. So there are many, many things that happen. So it's not to argue that the only thing that happens is in the brain. But if you look carefully on children with NF1, they have these different symptoms: seizures, headaches, brain tumors, brain-blood vessel defects, or problems with blood-brain barrier, learning disabilities, very high incidence of ADHD, smaller but also high incidence of autism, some incidence of mental retardation, and macrocephaly. So this is something that really piqued my interest. The reason why we thought it would be useful to look at this animal model is that this is an animal model because it's a single gene mutation, that the single gene deletion in the animal model mimics the behavioral phenotype and the pathological phenotypes very, very well. Okay, so went with that. So what are the symptoms? The first symptoms is café au lait spots. It's actually one of the things that show up in babies when they are very young, and it's a clinical indication to test for this condition. When they are older, there's these freckling in skin folding regions, these neurofibromas, plexiform neurofibromas on the skin. You can see it's also on the face, and there's a deformation of the bones and scoliosis. So many, many things happen. One of the major causes for morbidity are low-grade gliomas. Okay, so slow-growing tumors. They show up in the brain. If they are non-operable, they can be fatal. Many times they are operable and they are being removed, and it's a condition that can be managed. Okay, so here I'm kind of, again, dermal neurofibromas, plexiform neurofibromas, skin fold freckling, and learning disabilities. It's a two-hit model, so you have accumulation of these genetic mutations. So it means that while the café au lait spots show up very, very early, learning disabilities show up later in life, and we know that these effects kind of accumulate. Okay. So how did we study this condition? We essentially adopted the approach that was developed in Karel Svoboda's lab by Mao. There are others that are using this approach, that essentially looking at the projections from a target region, and we used that, the data from the Allen Brain Atlas, along with our own data, functional data, but using the same analysis. So rank ordering the amount of projections and looking across the two conditions, so healthy animal, littermates, and animals with genetic mutation. Okay. So this is our data. This is actually the Allen brain data, the labels, and the projection from primary motor cortex. So we can really quantify the amount of projections. We can rank order them, and we can look at our own data relative to that. This is already kind of the full list of projections from the Allen brain. They match the Mao dataset very well. So two different datasets with the same approach producing the same responses. And here is our data. Okay, so now we have, this is again the functional MRI data, putting a seed in primary motor cortex, looking at the projection of the functional connectivity data across the entire brain. So you can see here the clouded putamen. We can actually estimate the number of voxels that pass the statistical threshold. So again, it's a group data, and we can do this analysis for a wild type relative to NF1. Okay, so this is the two maps in the two animal groups, and we can rank order it. The slanted dashed lines means this is where the functional connectivity data overlaps the anatomical data. Okay, so the strongest projections in the anatomical data show as the strongest projections in the functional MRI data. You can see it here, and you can already notice that there's a difference between the wild type and the NF1. So for this is somatosensory primary, motor cortex secondary, contralateral motor cortex show decrements, and the clouded putamen, both ipsi and contralaterally, shows an increase in the number of voxels that show correlated activity between the NF1 and the wild type. So another way to do this analysis is to actually take just the ROI, regions of interest, based on the Allen brain atlas, so not count the number of voxels per label, but just look at these kind of maximum projection regions taken from the Allen brain and plotting the R value, or the correlated value. And this is essentially showing this, the data that you've seen before in a different way, showing the exact same response. So the cortical motor network showing a decrement in responses in the NF1 relative to the wild type, but the clouded putamen shows an increase. We looked at the somatosensory cortex, again doing the same analysis, and here what you can see is that the clouded putamen shows again an increase in these responses, but the primary somatosensory is also showing an increase. So again, if you look at the label, so this is the motor cortex, now looking at the primary somatosensory cortex, so looking at the projections from the Allen brain, what we see here is that the clouded putamen shows an equivalent response to the motor cortex, but shows an opposite response in the cortex. So here we see a decrement in activity, and here we see an increase in activity. Okay, so how can we explain this pattern of results? We went to the literature. This is from Anatol Kreiter's group, looking at anatomical projections using D1, D2 Cree animals to actually identify just the direct and indirect pathways of the basal ganglia. And what they've demonstrated in that paper is that there's a kind of a bias for the indirect pathway representation for the motor cortex, and there's a bias for the direct pathway representation in the somatosensory cortex, essentially explaining these opposite effects on cortical activity. So whether the output from the direct pathway, whether it suppresses or releases from inhibition the thalamic cortical projections. Okay, so the last thing that we've done with this analysis was to actually look at the spatial specificity. So another prediction that comes from the Wall et al. Paper is that the projections from the cortex, from the motor system and the somatosensory systems are going to be segregated on the anterior-posterior axis of the striatum. So this is optical density data, so anatomical projections data from the Allen brain. So again, not a Wall et al. Data set, a separate data set replicating the Wall et al. Response. This is in Bregma approximately -1. If you now plot the fMRI data, the resting state data, you can see that the reliable responses or correlated activity shows up anteriorly on the clouded putamen, overlapping these projections. If you plot the same responses for the NF1, you see this increased corticostriatal connectivity. If you do the same analysis for the somatosensory, primary somatosensory cortex, in the optical density, you now see a more posterior response, and you can see that the functional connectivity data shows up in the same location, and again, for the NF1, we get this increase. So again, showing spatial specificity. It's not that just in NF1, we got an increase everywhere in these correlated responses. We see increase that is specific to the maximal projections from that region. Okay, so the interim conclusions. It's actually, I haven't shown you that, but we have rotarod data, and now we are collecting data with animals doing a hand-reach task. We see these animals slower. On the rotarod, they fall faster from the rotarod, and the terminal speed is lower. With the hand grasp, they are slower to grasp. So we know that they have motor impairment, so that system is impaired. We saw that with the fMRI, we have increased somatosensory connectivity. We have increased somatosensory and motor cortices connectivity to the basal ganglia, so these functional connectivity measures. And a possible explanation is the differential modulation of the basal ganglia, direct and indirect pathways. So the next obvious thing to do was to image NF1 patients and do the exact same analysis, and we've done that. I'll show you these results quickly. We essentially look at the motor cortex and somatosensory cortex. TDC stands for typically developing children. We look at the responses in the caudate putamen, so I'll just show you these caudate and putamen responses. Putamen over here. And if you plot the NF1 relative to the typically developing children, again, we see, using the exact same analysis, increased responses in their corticostriatal measures, both for the motor and the somatosensory cortex. So again, arguing that this is an interesting result. For those of you that work with humans, we've actually compared the corticostriatal entire connectivity in these two populations across different networks. So what we see in the control, in the TDC, a majority of voxels within the caudate putamen correlated with activity in the control and ventral attention networks, and a much more kind of a splattered response in the NF1 children with a majority of voxels dedicated to the default mode network. And you can see it actually here that the responses for the typically developing children is very neat and orderly, and for the NF1 it looks like a very kind of messed up response. By the way, this is an interim stage in the development. We've seen that this matures to a slightly different state. So we know that this system is kind of undergoing organization at these ages, and it has clinical implications for treating these children with novel drugs that are coming to the market. So how can we kind of translate this to a potential mechanism? So we looked into the literature to see whether there are any evidence for the involvement of norfibromin in the striatum. And what we came across, so you remove norfibromin, you increase RAS, there's an increase in ERK1C. If you actually look at the involvement of ERK1C, ERK1C is critical for the localization of the potassium inward rectifying channels KIR2.1 from the Golgi to the membrane. If you look at the expression of KIR2.1 in the cortex of a mouse, there's a high dense expression in the caudate putamen, also in the dentate gyrus, very little in the cortex, a little bit in layer 1, and the dense projections to the olfactory bulb. So this is highlights this as a target. This is from a more recent paper actually showing that if you suppress the expression of KIR2.1, there's a kind of a resulting in medium spiny neuron degenerated dendrites. They lose innervation from dopaminergic neurons. These are also things that have been identified in these animal models, lower dopaminergic inputs in the striatum. It was thought that there's a problem with the dopaminergic cells, but these results actually suggest that there might be increased striatal excitability. We just now started to do the electrophysiology for that. These are medium spiny neurons taken from NF1 animals and control animals. This is really something that we started in the past few weeks, and this is initial data showing increased excitability of these neurons. So it's kind of promising. There are many, many things that we can do, and we actually propose them. We hypothesize that there might be increased striatal spiny neuron excitability. What we can do in this animal model is we can cross this genetic deletion with D1 or D2 animals, or the D1 and A2A animals, and essentially delete neurofibromin just from medium spiny neurons in the striatum, and ask, do we recapitulate the behavioral phenotype? Do we recapitulate the neural phenotype? So there's a tractable way to actually test whether this is a proper therapeutic target. It has been shown that these animals fare better with a MEK inhibitor, that the Cazorla group actually shown to rescue localization of KIR2.1 to the membrane. So there are many, many clues here that suggest that this is a viable target, a therapeutic target for treating NF1 children. Okay, so to summarize, we have shown that impaired corticostriatal is potentially linked to neurofibromin deficiency associated with brain impairments, and we now invest efforts to actually demonstrate causality, so demonstrate that we can localize this disruption. We know that there's increased incidence of autism in NF1 pediatric patients, so this is something that has been kind of suggested. It also kind of makes contact with observations in autistic children, individuals that exhibit dysmorphic striatal regions. There's a lot of electrophysiology data. The most recent one is a SHANK3 mutant that the lab of Bernardo Sabatini demonstrated to show increased spiny neuron excitability and corticostriatal hyperconnectivity. So again, a different animal model linked to the autism. We think that these things are linked. Okay, so in the next 5 minutes, I will tell you a little bit about our efforts to move into behaving animals. The idea here is really, this was something that we wanted to do from the start. We just used kind of also the spontaneous activity as a way to get there, is to actually have the animals perform tasks in the scanner, ask questions about memory performance, see the entire brain kind of responses, and develop targets for electrophysiological investigations and optogenetic disruption. We're finally getting there. So this is the animals that you've already seen, head posted, doing very simple collection of water reward. We now have the ability to do controlled whisking. So this is similar to Ehud Achisal's artificial whisking, just using optogenetics. So we use redshifted opsins injected to the facial nucleus, and using a non-invasive lead that is placed outside the ear of the animal, we can control whisking. So this is something that we've just done. This is the fMRI activation that we get from something like that. This is the M1 projection and the whisker barrel field S1 projections. And this is the overlap between the fMRI and these two networks of regions, again showing that we're seeing the predicted regions from the connectional anatomy. This is our prototype for virtual reality MR-compatible MRI. The animal is on a treadmill here and walk actually forward and backwards with this treadmill. They actually like it very much. It's something very natural to them. It also makes them less stressful. This is the cradle without the components on top of it, but we actually measure the location of this treadmill, so we can change visual stimulus. This is the cradle. The sound is just the clicks for providing a water reward. There's actually here a sniff detector, so a cone that we can use to detect sniffs of the mouse. Sorry, I'll show you that. Okay, one component before last. This is our animals. Again, this is an MR compatible. So we have a projector. So this was a videotaped outside of the scanner. We can't do the videotaping in the scanner, but we have a projector and a screen in front of the animal, and now the animals learn how to do simple detection of visual stimulation. So that allows us to do calibration of the system. And the last component is the MR compatible olfactometer. So that was done with the assistance of Dima Rinberg. So this is an 8-channel olfactometer. This is the MRI. The trick here was to use essentially a non-invasive sniff detector. So the animal's nose is inside this cone, and we can measure the sniffs. This is actually measurements from the MRI, and we can align the presentation of odors to the sniff cycle. We can do the leak detection to see that the animal essentially responded to the stimulus. Just showing you a snippet of the first set of results. So just passive olfaction, olfactory MRI for 2 odors, showing a reliable response. So this is one of the kind of strongest stimuli that you can use. Responses of the animal behaving, so very simple water reward and looking at the brain signals for reward. You can see VTA, amygdala, globus pallidus, nucleus accumbens. Again, this is once you move into functional MRI and you do tasks, even for an 8 minutes run, you can get very reliable responses. So this is something that we are very happy with, and the rewards. Okay, so I hope that I was able to convince you that intrinsic functional connectivity can allow us to do comparative analysis across species. We've shown what we think is evidence for rerouting of information between mice and humans in the hippocampal memory system. In the NF1 animals, we actually observed a deviation of their corticostriatal connectivity, with hints that what we are seeing is a pathology of increased excitability of the medium spiny neurons. We think it might have to do with KIR 2.1, with the potassium inward rectification. There's actually therapies available that can essentially alleviate the ERK1C overexpression. So if this is actually the case and it helps, it can be promoted to a clinical trial, and we are actually in the process of doing that. Okay, I'll point out the people involved in the Eyal Bergman in the cortico-hippocampal connectivity work, Judd and Ben Shofty. Ben Shofty, this was his thesis, the NF1 animals and human data, and Judd joined that project. The other lab members, the lab manager, Sasha Kabushansky, Nadav Cohen, who's our colony manager, our collaborators at Tel Aviv Sourasky Medical Center, Shlomi Konstantini, Liad Ben Sira, Daphna Ben Bashat, at Cincinnati, Nancy Ratner, which we are now developing drug targets with her and testing them on the animals, Jackie at the Technion, Dima at NYU, and this is the students in the MRI, and thank you.
And he's dropping things. What does it mean in that sense that you are representing here?
So,
Kirk elevator?
No. So I think that this is like what we are seeing is essentially a disruption. It's actually, in my mind, it's in the electrophysiology data that we have, is a very mild disruption in kind of the excitability of these neurons, and it means that if the motor cortex is activated, the neurons are firing, it's going to kind of result in more activity in the striatum, and then suppression of activity through on the thalamus. So actually an overall net negative response in the cortex. For the somatosensory, so for touch, it means the opposite, right? Release from inhibition of the thalamus and more activity in the cortex. These children, by the way, they demonstrate slower motor responses, So their motor system seems to be kind of slowed down, and they have hypersensitivity to pain and to somatosensation. So we know that they are in a situation of hypersensitivity to touch. So these things, this is why, I haven't shown you the preliminary data for that, but this is actually something that were reported in these children. In the children, it was ruled out that the motor deficit is of cerebellar origin. So it was that was ruled out. So really what we think is that these neurons are in a slightly more excitable state because of this disruption in KIRS, and that results both in these kind of opposite responses on the cortex, but also it results, and this is something that I haven't shown you here, in disrupted corticostriatal organization. So we think, so this is something that is long-term, and we think that that's related to the ADHD and the autism high incidence rates in these children. So if you look at I've shown you in the humans, children that are between the ages of 7 and 11. If you look at the normal children that are about 17 or 18 years old, adults, they're kind of the corticostriatal connectivity looks different than what we see in the typically developing children. So the system is still changing, and this kind of messed up picture that we see in the children, now we have data from adults with NF1, it looks to be consistently kind of messed up. So what we think is that if there is a kind of possible way to kind of reduce the activity in the striatum, this is something that can be treated when they are still young and maybe lead to a normal development of this system.
But no, what I was meaning is normal people. You have two situations. In a battle, you don't feel anything, and you are very competent.
Yeah.
In an awkward situation, you are incompetent and you are very excited. So maybe this can explain a much more general.
No, so I don't think this is it. I think that the level of arousal that you're talking about is not going to operate through that system. It's going to be something completely different, a different network.
Autism has nothing to do with an extreme.
I don't think so. But,
okay.
Yeah, Eli.
I was wondering about the picture, the movie that you showed at the beginning of spontaneous activity, ongoing activity in mouse and man.
Yeah.
The question was about the spatial extent of the response. The spatial extent in the human,
Mm-hmm.
the spot in the frontal cortex was probably larger than the whole brain of the...
Yes, it is.
So my question is, what determines the spatial resolution here? There must be something about the spatial space constant of the population.
So it's an excellent question. What we do is in the sequence that we are measuring, we are kind of using spin echo imaging, which is kind of suppressing signals from large blood vessels and going more towards the diving arterioles and maybe capillaries. So this is a way to actually constrain the spatial resolution, and then we are kind of getting closer to, I guess, kind of the diving arterioles, the distance between them and spatial resolution. There's actually been just recently a paper in Nature Methods from the lab of Anatol, I'm blocked on the name. Xin Yu is the first author, showing a single vessel responses with fMRI, so in the mouse. So actually showing that they can detect these responses that are very well localized. So diving arterioles. So they looked at diving arterioles and they show a voxel that is just above the diving arterioles, and they're showing kind of a single vessel response. Now there is a question about whether the resting state, these intrinsic spontaneous fluctuations, are localized to that level. I can't answer that question. I just know that with these types of responses, so for example, the projections to if we look at the caudate putamen and the spatial distribution, so on the order of a few hundred microns, we do see segregation according to networks. Okay, so if I actually say, okay, with the injections that the Allen brain used, the data that we have is on an equivalent spatial resolution. So what they can do, we can do. Whether we can do better than that, what they've done, I don't know. So I would say, if you would ask me to put out a number, I would say on the order of a few hundreds of microns. So 200, 300 microns. In humans, it's possible that with high field, people will report similar kind of, maybe not a few hundred microns, but sub-millimeter, which I think was done by Rainer Goebel. Yes, Aviv? It's the same frequencies, okay? We haven't seen a shift in the frequency. What we don't have is we don't have the overlap of the physiological signals because the mice are breathing faster and the heart rate is faster, so even the harmonics don't fall into the, we don't have aliasing into this spatial resolution. So it seems like the frequency is not shifted, at least in our hands. But we haven't done, like, you know, like really trying to see whether the peak is different. Yes.
That's last question I want to ask. So you were monitoring both the hippocampus and you are now giving orders, and I was wondering because there is the other function, there is a strong notion that a strong connection between function and memories and the hippocampus. I was wondering whether you look at functional connectivity between these areas.
So we do. We have data. We are doing the task. It's just that it's too early to report on that. But this was the motivation to do olfaction. It's a very kind of strong way to manipulate. The animals learn to do detection for disorders really, really fast. So it's really a fast way to do memory experiments, which was. Sorry, I can't be more.
Okay.
Yes.
Very last question.
Yes, please.
Do you have any information about how many neurons do you need to decide to read and be connected?
No, not really. There's a very, like a fundamental question of what drives these responses, and there's a kind of nowadays kind of an argument between a few individuals in the field claiming that they know what is driving that response. I don't know if they didn't I didn't see them estimate number of neurons, but they talk about capillaries essentially vibrating at the gamma frequency and generating that response. You know, the jury is definitely out on whether that's true or not, but they're very strong and considered. Anyway, we'll see.
Okay, thank you, speaker.
Anything about mice being evolutionary, more advanced or closer to man than rats?
No, if anything, it's the opposite. They're considered further.
Somebody made a claim here.
Where? Who?
Here,
I'd love to meet you.
You'll have to meet him. Well, he can tell me. It was 3 lectures ago or something like that. Actually, so why were you doing it on mice and not on rats?
Because of the genetic models.
Meaning?
We would like to do genetic manipulation, like the NF1. All of that work is done on mice.
So just completely.
It's like a field, I guess, excessive method.
This is really something. I mean, you mean that in the rats, the brain of different species functions like humans?
We're actually not the first to show that. There's been arguments about previously.
And it has implications for deformation or in diseases where some parts are excited and
קצת על השעון, אני חושב שהשעון היה קצת יותר מוקדם. זהו, זה מה שאני אומר, שיכולתי להעט ולא... מי סיפר לכם שרעצם יותר חוקים מבני אדם לעומת הסלובניה?